Welcome to the 22nd lecture of this NPTEL course on environmental biotechnology, which is on the physiological ecology and resource exploitation by microorganisms. In this particular lecture, the complex mixture in chemical disequilibrium in the environment will be discussed, and this will be followed by the thermodynamic hierarchy that describes the selective pressure, energy sources and geochemical reactions within different environmental systems. So, to begin with, the complex mixture in chemical disequilibrium in the environment. So, one of the major aspects of cellular metabolism within diverse environments, wherein environmental biotechnology processes are studied or developed, generation of ATP, the energy currency of the cell, remains one of the very important factor for driving the cellular processes. And as we understand that this generation of ATP, which is the energy currency of the cell, is connected to metabolism, cellular metabolism with respect to deriving the energy from the different energy sources like the chemical energy or the light energy, or it could be from the reduced carbon substrates or carbon compounds or the sugar molecules, which upon oxidation also produce this energy or the ATP molecules. Now, without these ATP molecules, cell maintenance, cell motility, biosynthetic reactions which facilitate the synthesis of all the macromolecules within the cells, replication of the DNA or the genetic material, cell growth and heredity would be impossible. Now, the biochemical mechanism of ATP generation fundamentally relies upon either the substrate level phosphorylation, or it could be through the membrane bound electron transport chain or electron transport system. Now, as we possibly understand that the substrate level phosphorylation which occurs during the glycolytic reactions of the metabolism, particularly when the sugar molecules are oxidized through Embden-Meyer or Parnes pathway, and we observe that there are steps through which the ATP molecules are generated, and as the sugar molecules are broken down into smaller molecules like the pyruvic acid, for example, from the glucose. At the same time, these catabolic reactions like the Embden-Meyer or Parnes pathway or the glycolysis also produce the reduced electron acceptor, that is the NADH or the electron carrier, and the pyruvic acid which is produced upon oxidation of the glucose molecule is further metabolized through tricarboxylic acid cycle or TCA cycle and leads to the production of further NADH or reducing powers. Now, while this ATP which is directly produced from substrate level phosphorylation represents the one of the major outcome of this glycolytic reaction, the reduced electron carrier like NADH, which are produced in plenty of the number during this glycolytic reaction and subsequent TCA cycles, they transfer the electrons into the electron transport system, and as you can see in the right panel that the reduced electron carriers like NADH or FADH2, which are produced during the glycolytic reactions or the TCA cycle, they donate the electrons to the electron carriers which are membrane bound, and these membrane bound electron carriers, they are able to transfer the electrons to the terminal electron acceptor like the oxygen, and thereby producing water molecule. But more interestingly, while the electrons are transferred from the reduced electron carrier like NADH to the terminal electron acceptor oxygen, during this electron transport system, some of the electron transporting complexes also act as proton efflux and efflux pumps, and they effluxed out proton into the periplasmic space or the cell exterior. Now, this electron transport chain, this is called electron transport chain, the transportation of the electrons from the reduced electron carrier like the NADH or FADH2 to the terminal electron acceptor, and this represent the electron transport chain, which is composed of different electron transporting carrier complexes. Now, due to the functioning of these proton efflux process or proton efflux pumps, which are integral properties of the sum of the electron carriers carrying electrons during this membrane bound electron transport chain, these create the proton or sodium proton motive force. So, essentially we see that a large number of protons accumulate within the periplasmic space and thereby creating the proton motive force, and this proton motive force essentially then drive the synthesis of ATP through the enzyme complex which is known as ATP synthase, which is embedded into the inner membrane or the cytoplasmic membrane. This picture elaborates this process, that the complexes, as they carry electrons from NADH to oxygen, they exports the protons, and these protons are allowed to come inside through this ATP synthase complex, enzyme complex, and this ATP synthase complex essentially generates the ATP molecules which are required for the cell. So, essentially what we understand, that the oxidation of the substrates, either the energy source or the carbon source which are reduced, which can be utilized as both energy and carbon source, the electrons are derived, and those derived electrons are given to the oxidized NAD to produce the NADH, and the NADH carries the electron to the electron transport complex, which finally leads to the production of the ATP through the proton motive force. Now, the ATP synthase enzyme complex, which is responsible for the production of ATP, the activity of this ATP synthase enzyme relies upon the formation and maintenance of the electrochemical potential across the membrane. So, this membrane must be intact. So, within an environmental context, we need to understand that the membrane integrity must be maintained all time during the cellular process. Any toxic substance or any toxic condition which might interfere with the cellular membrane constituents or disrupt the membrane, the cellular integrity, membrane integrity is compromised or lost, might or will definitely result into the production of this proton motive force, and essentially which will lead to the decreased production of the ATP from those oxidation reduction reactions. Now, the ultimate driver of the ATP synthesis is the variety of thermodynamically unstable materials that are commingled in the water, sediment and soils within the biosphere, or basically any part of that biosphere, that is the environment. Now, what does this mean? We have just seen that the ATP production is actually facilitated by the oxidation of NADH or the reduced electron carrier. Now, where from these reduced electron carriers like NADH are produced? The NADH are produced because of the oxidation of the substrates molecule. What type of substrate? Substrate which are energy rich, and upon oxidation they give rise to the electrons, and those electrons are carried by the reduced electron carrier or the oxidized electron carrier to produce the reduced form, that is NAD plus is converted to the reduced form of the NAD plus, that is the NADH H plus. Now, who are those electron donors? The fundamental electron donors from where the source of the electrons, from where the electrons are originally obtained and then taken by the NAD plus. Those sources are the thermodynamically unstable materials. For example, the reduced carbon compounds or other inorganic materials which can be processed or catalyzed or rather metabolized through microbial enzymes to get the electrons out of them, and then those electrons can be given to the electron carrier like the NAD plus. Now, these materials, for example ammonia or reduced sulfur compounds or iron oxide compound, all these compounds are mixed in a very active form within diverse components of any kind of environment, be it wastewater or be it a rock or a sediment or a polluted soil or any kind of other materials. So, everywhere these materials, which are basically many of them are thermodynamically unstable and could be metabolized by the cells to get the electrons out of them most often, and sometimes donate the electron to them to reduce them, and that drives the synthesis of ATP. Now, let us look at the different subsets or sub-habitats within the planet Earth. So, the Earth as a whole or its different habitats or sub-habitats and their microenvironment, the innumerable number of microenvironments which exist within this landfill dump site or a pile of ore material, mineral containing or heavy metal containing rock ores, or a agricultural site or a activated sludge containing wastewater treatment plant. So, we can have innumerable number of microenvironments within them, and they constitute a kind or represent a kind of complex state consisting of commingled material. Now, these commingled materials are, most of them are organic or inorganic substances. These organic or inorganic substances are active in the term that they can donate electron or they can accept electron. Sometimes they can be oxidized like a carbon source, like a alkane chain or a glucose molecule or any other reduced organic carbon which may be oxidized, or it may be ammonia or sulphide or heavy metals which can be transformed through redox reactions. So, these materials are all mixed together in any kind of environment. So, wherever we try to implement or investigate environmental biotechnology processes or try to develop environmental biotechnology processes, be it for increasing the productivity of the crops or handling some of the environmental pollutants or making the water treatment process more effective. Now, these all environments are in a state of heterogeneous mixture, and most of the time these mixtures represent mixture of rock, water, gas and other materials, but in sunlight. Now, what is the significance of sunlight over here? The sunlight drives the photosynthetic reaction most of the time. Under dark condition also, the fixation of carbon is possible by many microorganisms which are chemoautotrophic microorganisms, but principally on the surface of the earth or any environment which is exposed to sunlight, this light driven photosynthesis or fixation of the inorganic carbon into reduced carbon material plays a very important role by providing the reduced carbon substrate. Now, this reduced carbon substrate makes a very important position in entire thermodynamic process that drives the metabolic reactions, because these are readily oxidizable. They can be oxidized provided the condition is suitable and the appropriate enzyme complexes by the microbes are available, and they can produce electrons. These electrons can be given to NAD plus to produce NADH plus, and NADH plus can create the proton motive force, and from the proton motive force the ATP molecules can be generated. Now, it is of fundamental importance to know how do microorganisms make physiological sense of this complexity, because they, in every environment or a part or within a micro environment, micro habitat, there are, as I mentioned, the innumerable types of substrates are there. So, this is perfectly a kind of a heterogeneous mixture of different oxidized and reduced substrates. Now, microorganisms need to make living out of that. So, we need to understand how they make physiological sense. Now, in order to understand that how microorganisms make a physiological sense out of that, we need to follow certain principles of thermodynamics. Thermodynamics, which is one way to achieve a unified and orderly view of the biosphere or a part of the environment. It is also a branch of the chemistry that rigorously predicts the chemical reactions that are energetically favorable and ones that are not. So, looking at the thermodynamics, we can understand the reactions, which reactions are favorable and which reactions are not favorable under certain condition or under standard conditions or biochemically or physiological conditions also. Thermodynamics can systematically arrange the types of chemical reactions. So, if you have a number of chemical reactions which are possible, as I mentioned earlier, oxidation of water, oxidation of ammonia, oxidation of nitrite, oxidation of sulphide, oxidation of glucose, and simultaneously reduction of oxygen, reduction of nitrate, reduction of sulphate, reduction of iron. So, many reactions are possible, which are, I am talking about mostly the oxidative and reductive reactions. So, many reactions are possible in an environment. Now, is it possible to arrange the type of reactions that could perhaps occur within a complex heterogeneous mixture? based on certain criteria? Yes, it is possible. So, it is the thermodynamics or thermodynamic criteria or characteristics which help us to arrange the type of reactions, particularly the oxidation reduction type reactions that may occur in a complex heterogeneous mixture, which are typical for any environmental system. And when we have that arrangement, that the reactions which are more preferable in an oxidizing conditions and reactions which are more preferable within a reduced conditions, we can predict and catalog the energetically favorable reactions. We can pretty easily identify which type of reactions might be more preferable or energetically preferable or favorable within a given environment. So, this is a very common plot that often we use to identify these thermodynamic criteria, that the reactants are converted to products and we can calculate the free energy change, and the types of reactions can be of different nature: change in state, dissolution and precipitation, complexion, acid base reaction, sorption desorption type of reaction, and oxidation reduction reactions, which are very relevant for biological systems, particularly of environmental relevance, to generate ATP. However, sorption desorption kind reactions, complexion, dissolution, precipitation, and even the changes in state are also important with reference to change in the different type of environmental pollutants or other compounds which are useful or relevant for microbial metabolism. Now, with the help of the thermodynamics, we can identify resources which are exploited by microorganisms as their energy sources and facilitate the ATP generation. So, if in a kind of an open landfill site, if we have certain substance, we can identify these substances by some chemical analysis, and looking at the redox condition, that is the oxidizing or reducing condition of the environment, we can very well identify or define which are the substrate which might be or possibly be oxidized by the microorganism under the given environment, or what are the compounds or what are the substances which might not be metabolized, like oxidized or reduced under this given condition. It is the thermodynamically unstable resources particularly that have provided the selective pressure for microbial energy production strategies throughout the evolution. Like for example, if we look at the oxidation of the water, the breaking of water, the photolysis, which as we know during the photosynthetic reaction. So, why photolysis of water is so preferred compared to other? Perhaps that is more favorable thermodynamically compared to any other reaction. Why reduction of oxygen to water is considered to be energetically more favorable rather than reduction of sulfate to sulfide or reduction of carbon dioxide to methane. So, in an environment, if we have both carbon dioxide and oxygen, for example, now are we going to have methane generation from carbon dioxide? Possibly no, because the reduction of carbon dioxide to methane is energetically not favorable. Thermodynamically, it is not favorable. The free energy change, the delta G, is very, very less. So, the microbes will not be benefited. So, if we have oxygen or any other terminal electron acceptor which are having a better free energy provision, carbon dioxide reduction to methane may not be adopted by the cells over there. So, even if you have organisms, microbes who are able to carry out methane generation from carbon dioxide, those microorganisms won't be acting appropriately towards the generation of the methane. Now, in the next point, we are going to discuss about the mechanistic relationship between oxidation reduction processes and the ecosystem function. As we have understood that photosynthesis is one of the major driving process in this entire biosphere and also in any kind of environment. It is not only photosynthesis, but it could be chemosynthesis also, like chemoautotrophy. So, photosynthesis is very important because the plant biomass that we see, or the algae and the phototrophic microorganisms who absorb the light energy and can fix the carbon dioxide into the reduced carbon. So, basically with the help of the light energy and with the available carbon dioxide and water, the reducing power like the NADH are produced or NADPH are produced, and at the same time the photolysis or the breakdown of the water molecule leads to the production of oxygen. So, this has a very important implication on the overall the metabolism or metabolic scenario. This reducing power is used by the microorganisms and other organisms who are carrying out the photosynthetic reaction to reduce the carbon. So, essentially the carbon dioxide is reduced to reduced form, like the, for example, the glucose or glucose like reduced carbon form. And these reduced carbon molecules, they allow or they represent the primary production within an ecosystem. So, in any kind of ecosystem which are exposed to solar light, we can expect that the primary production is by the photosynthetic reaction. However, in environment where sunlight is not received, the primary production could be manifested by the chemoautotrophic reactions, which are microorganisms which are capable of fixing carbon dioxide, the same reaction, but without the involvement of light. The energy is obtained from other organic or inorganic substances in that case. So, we have already seen that there are different type of microorganisms which are maybe the lithoautotrophic or organoautotrophic organisms who can obtain the electrons from either the chemical forms like the inorganic chemical compound or the organic compound, and utilize that electrons to fix the carbon dioxide to reduced carbon, and essentially that reduced carbon forms the primary production. So, no matter whether it is the photosynthesis or it is the chemosynthesis, this reduced carbon is going to form the base of the primary production. Now, this synthesis of this reduced carbon in all ecosystems, all environments, that creates the pool of both oxygen and reduced carbon in the biomass. So, this reaction has a very important bearing on the environment, because it produces both the oxygen as well as the reduced carbon. So, if we look carefully, reduced carbon could be the substrate for the heterotrophic or the organotrophic organisms. So, any organism who is not capable of fixing carbon dioxide would rely on this reduced carbon and metabolize this carbon to or oxidize this carbon to produce the necessary ATP molecules and also the precursor for the macromolecular synthesis and other cellular metabolism. On the other hand, this oxygen which is produced out of this photosynthetic or chemosynthetic reactions could actually take part in the electron accepting reaction. Some time ago we were looking at the electron transport chain. So, we have also noticed that within the electron transport chain there is a terminal electron acceptor. The electrons which are basically subtle to the electron transport chain must be accepted by some acceptor, and after that or beyond that no more electron transportation reaction will occur. So, that is called the terminal electron acceptor. So, oxygen is the most preferred terminal electron acceptor. Reason behind that, the free energy gain from the by reducing oxygen to water molecule is found to be maximum within the biological context. And this formation of oxygen and reduced carbon in the form of biomass assures a state of disequilibrium, because out of the carbon dioxide and water and other inorganic resources, it is continuously producing the reduced carbon molecules. So, this formation of carbon molecule and the oxygen, both that assures the state of disequilibrium. Now, photosynthesis or the chemosynthesis of the fixing of carbon dioxide is very effectively reverses the universal drift toward thermodynamic equilibrium. We are going to talk about that very soon. Now, overall what we have understood that that the oxygen molecules which are generated and the reduced carbon which are produced, they basically drive the microbial metabolism through a series of oxidation reduction reactions. Now, the major oxidation reduction reactions which are responsible for the drift are the metabolism by the chemosynthetic organotrophic life form, and the other one is the metabolism by chemosynthetic lithotrophic life form. So, in the chemosynthetic organotrophic life form, blending of reduced biomass, the reduced carbon which is produced. We just saw that following carbon fixation, the reduced carbon are produced. So, that reduced carbon, when it mixed with the oxygen which is present, because oxygen is also produced, oxidized compound, because in any environment there will be plenty of oxidized compounds including nitrate, iron oxides, manganese oxide, sulphate, carbon dioxide, etc. that sets the stage for metabolism by organotrophic life, because organotrophic organism would actually oxidize the reduced biomass and use the oxygen as the terminal electron acceptor. Sometimes these are also used as their other electron acceptor if oxygen is not available. On the other hand, the lithotrophic life form will rely on the commingling of the reduced inorganic substance, because there would be some substrates which are always inorganic substances which are always reduced, like the ammonia for example, or sulphide for example, or even hydrogen. So, these reduced inorganic compounds with oxygen, because again oxygen would be there most likely, and other oxidized materials set the stage for the metabolism by the chemosynthetic lithotrophic form. So, in any environment, or it is a part of the environment, we will expect that this organotrophic and lithotrophic form of lives are both possibly running parallelly. Maybe sometimes the organotrophic life will predominate over the lithotrophic life. Some of the environments we see the lithotrophic life is more abundant or more dominant over the organotrophic form, but it is highly likely that the reduced biomass eventually produced by even the lithotrophic mechanisms would be metabolized by the organotrophic life form. So, overall a non-equilibrium state is created by co-occurring pairs of the reduced and oxidized materials in water, sediment and soil of the biosphere. For example, oxygen, water, nitrate, nitrogen, MnO2 to Mn, nitrate to ammonia, sulphate to sulphide and carbon dioxide to methane. Now, next we will study that how the use of this thermodynamic hierarchy can be made, particularly with respect to the Hopf reactions, to predict biogeochemical reactions in time and space. And this particular thermodynamic hierarchy that we will see now is considered as a compass for environmental microbiologists and environmental biotechnologists, because it helps in predicting and interpreting microbiologically or microbially mediated ATP generating reactions. So, relationship between reduced and oxidized substrates as a vertically arranged hierarchy of oxidation reduction Hopf reaction. So, we can see there are multiple Hopf reactions are presented over here, and the electron volt is mentioned, and the PE is also presented as at pH 7. So, the compounds which are listed on the scale on the left side, we have the compounds which are in their oxidized form, for example, oxygen or the nitrate or the nitrite or MnO2, these are all oxidized. And on the right hand side on this particular scale, we have the compounds which are in their reduced form. Basically, these compounds can be acted as an electron donor. So, for example, water, H2O can be an electron donor. Similarly, if we have ammonia, ammonia could be electron donor and so on. Now, the transition from this oxidized state to the reduced state depends on the redox status of the system of interest firstly. Now, whether oxygen will remain as oxygen or will form water, or nitrate will remain as nitrate or form nitrogen, dinitrogen or ammonia, or for example, the selenate will form as a selenate or selenite, all these things depend, or MnO2 will remain as MnO2, will form MnCO3. These all depends on the redox status of the system of interest. Firstly, that what is the redox condition, because this is a scale which is considered as a compass. Now, using this compass, if I go to a landfill site and if I go little deep inside the landfill site, like I dig maybe 1 or 2 meters inside the landfill site, I may encounter a reduced condition or a oxidized condition. So, the kind of reactions which would be possible at that state, because the redox condition or the redox state of the system is going to control that what type of reactions, whether these acceptors will be accepting electrons and converted to electron donors or not, or vice versa. So, if we look at the status of these conditions, we will find that some of the reactions are favorable under highly oxidizing conditions, as you can see from these PE values, while some reactions like reduction of carbon dioxide to methane, for example, or reduction of sulphate to sulphide, these are preferable or favorable under highly reducing condition. So, these are the two things. One, we need to assess what type of Hopf reactions are possible, that is the substrates which can be oxidized or reduced by different analytical techniques. We can take any wastewater or contaminated site or any environment where some biotechnological processes are to be initiated. So, firstly we need to analyze that what are the substrates which are the unstable substrates which can be oxidized or reduced. So, potential electron acceptors and electron donors are to be identified, and then it is necessary to identify whether the environment is a reducing condition or an oxidizing condition, because some reactions might be possible under oxidizing condition, but some reactions might be only possible under the reducing condition. I mentioned about the formation of methane from carbon dioxide some time ago, which is possible only under highly reducing condition. So, if we see that high amount of carbon dioxide is there, but the condition or the redox state is positive, like positive 10 or 5 or 6, then we may not expect any kind of carbon dioxide reduction under the same condition, because under that condition oxygen is going to be reduced and to produce water, or if oxygen is lacking, then eventually nitrate, after nitrate Mn or maybe iron will be reduced, but carbon dioxide reduction would definitely not take place unless the condition is going to be highly reduced. Now, this compass can be used to predict which combinations of the half reaction pair are thermodynamically possible. These are all reactions from these thermodynamic the half reactions. So, the half reaction pair, out of them which one are possible in a given environment is only predicted from this compass or the list. Under standard conditions, the lower reactions proceed leftward, that is electron producing, and the upper reaction proceeds rightward, that is electron accepting. So, under normal standard conditions, we will see these reactions will prefer, like the oxygen would be reduced to water, or nitrate will be reduced to nitrogen, etc. Whereas, under the reduced condition, we may see that carbon dioxide is reduced or sulphate is reduced to sulphite. Now, if we look little more in depth into this particular compass, we will be able to find out that this is very well connected to the carbon cycle as well, or in any environment where reduced carbon or carbon molecules are cycled. In all environments, eventually carbons are cycled. So, fundamental reactions to the carbon cycle tie the oxidation of this photosynthetically produced or otherwise produced organic matter to the variety of final electron acceptors that may be present. Now, if we look at the carbon or the glucose, this can be oxidized. So, this can be oxidized to produce the electrons, and these electrons will move into different electron acceptors, like it will move to oxygen as a terminal electron acceptor. The electrons might move to the nitrate or to the sulphate or the selenate or arsenate or even to carbon dioxide to produce methane. Now, the free energy change or the free energy gain by this transfer will vary, because if the electrons travel from this glucose oxidation to water, the free energy gain is maximum compared to that of nitrate or selenate or arsenate or sulphate or carbon dioxide. So, essentially we can expect energy gain which decreases very significantly as we come down in this particular thermodynamic scale. Now, each of these coupled half reactions is mediated by chemosynthetic organotrophic microorganisms. So, the reactions that we are mentioning, like the reduction of oxygen as a terminal electron acceptor, reduction of nitrate to nitrogen, or reduction of nitrate to ammonia, or reduction of selenate, or reduction of iron oxide, arsenic, sulphate to sulphide, or carbon dioxide to methane, are all catalyzed by specific group of microorganisms. Sometimes some reactions are catalyzed by more than or a single microorganism can catalyze more than one reaction, but often they are very specific to individual group of microorganisms. So, based on this overall process, we could find out 8 common processes. These are which are recognized to occur in a carbon rich habitats, which includes the aerobic respiration to denitrification, manganese reduction, iron reduction, fermentation, sulphate reduction, methanogenesis, and finally acidogenesis. So, as I discussed, aerobic respiration will occur at the PE of 13.75 or nearly 4, because it is the most oxidizing condition which prefers or favors the use of the oxygen as a terminal electron acceptor, and its free energy yield exceeds that of the other reactions. So, if you have the free energy content is the maximum free energy constant, that is the minus 125 kilojoule per reaction. So, that is the maximum. So, if we have oxygen in a system, oxygen is going to be use a terminal electron acceptor if the condition is oxidizing. If we have to look at the PE also. If the condition is not oxidizing, then perhaps oxygen may not be reduced. Now, in a given aquatic or terrestrial habitat rich in carbon, microorganisms endowed with the physiological capacity to carry out aerobic respiration will obviously have an advantage. So, it is not only the aerobic respiration which will provide more energy to the system or the cells, but also the organisms which are capable of performing the aerobic respiration will have an advantage, and certainly they will proliferate, they will make use of this energy more. Their ATP generating ability exceeds that of the other microbial residents, and as long as the supply of oxygen is adequate, aerobic respiration will predominate. However, as soon as the oxygen level declines, because the oxygen level, if it is a kind of an environment where the free diffusion of oxygen is not possible, then the oxygen level is going to be declined because the organisms are continuously using the oxygen. Then as the oxygen level declines, because oxygen is continuously used as a terminal electron acceptor, the nitrate respiring organism will become predominant. Why nitrate reduction to denitrification process? Because its energy yield is the next to oxygen and it is higher than the others. And once the nitrate is also exhausted, that is the denitrifying process is done, these microorganisms eventually proliferate for some time, and then the subsequent processes like manganese, iron reduction followed by fermentation and other reactions will follow. Now, this predictable sequence of physiological processes is reinforced at the level of gene regulation within individual microbial cells. So, it has been found that final electron acceptor allowing the highest free energy yield inhibits expression of genes required for utilization of final electron acceptor residing lower in the hierarchy. For example, if a particular organism, maybe a Pseudomonas strain, is having both the genes for aerobic respiration as well as for the denitrification or maybe for manganese reduction, these genes are differently controlled. So, in the presence of oxygen, the denitrification related gene or the manganese reduction related genes are going to be repressed. That is very important. Some bacteria, as I mentioned, can perform more than one electron transport processes. In natural environment, many times many bacteria are capable of doing this. They are capable of utilizing multiple electron acceptors, and many of them might have a branched electron transport system as well. However, it is highly regulated by the genetic background of the cells. As I mentioned, the genes which are involved in reduction of other terminal electron acceptors, which are provided with or will lead to the production of less amount of energy, will be repressed unless the maximum energy yielding process is exhausted. Those genes are not going to be expressed. So, with this we end this particular lecture, and this part of this lecture may be read from the environmental microbiology book From Genomes to Biogeochemistry by Madsen. And in conclusion, any part of our environment represent a state of commingled materials. How do microorganisms make a physiological sense of this complexity is an important aspect of environmental biotechnology. Thermodynamic logic in utilizing the resources and generation of ATP is discussed. Mechanistic relationship between the oxidation reduction processes and ecosystem function is highlighted. And finally, use of the thermodynamic hierarchy of high reaction to predict biogeochemical reactions in time and space is discussed. Thank you.